Hello, I'm Dr. Shen-Gunther, Chief of the Department of Clinical Investigation at Brooke Army Medical Center. We're so excited and grateful to have Dr. James Allison join us today and share his work. To truly appreciate the significance of Dr. Allison's work, I will begin by telling you a brief story about cancer.
In Charles Graeber's book, The Breakthrough, Immunotherapy, and the Race to Cure Cancer, the author writes, "We have had three main methods for treating cancer. Surgery for at least 3,000 years. We added radiation therapy in 1896. Then in 1946, chemical warfare research led to the use of a mustard gas derivative to kill cancer cells. The cut, burn, and poison techniques have been the foundation of cancer treatment, until now." Before I introduce Dr. James Allison, our keynote speaker, let us take a brisk walk through history. Cancer was first described by the ancient Egyptians about 3000 BCE, or 5000 years ago. The papyri manuscripts shown here revealed that Egyptian physicians were skilled surgeons. They removed superficial tumors using the knife or hot cautery. However, for internal tumors, such as cancers of the stomach and uterus, they did not dare operate. Instead, they used medicinal compounds such as barley, beer, and pig's ears. Surgery remained the only remedy for 5,000 years. During the Renaissance, Ambroise Paré became the greatest surgeon and medical writer of the times. Paré was an unschooled French barber surgeon, who apprenticed under this brother, also a barber surgeon. He received his surgical training in the army on the battlefields, and wrote 10 books on surgery, 1564. On cancer, he recommended surgery only if the cancer could be totally removed, recognizing the futility of incomplete surgical resection. 300 years later, radical surgery proved to be ineffective, and the only treatment for early cancer. Here, Dr. William Halstead of Johns Hopkins University achieves the first successful results of radical mastectomy for breast cancer, in 1894. The following year, in 1895, Dr. John G. Clark under the direction of Dr. Howard Kelly, also at Johns Hopkins University, achieved curative results with radical hysterectomy for early cervical cancer. The discovery of radioactivity ushered in the era of radiation therapy. In addition to their numerous discoveries, the Curies described how tumor cells were destroyed faster than healthy cells after exposure to radium. In 1896, Dr. Emil Gruber became the first physician to apply X-rays as a treatment for recurrent breast cancer, like this photo taken in 1903. Then came chemotherapy. It is ironic that the first effective anti-cancer drug came from mustard gas developed during World War I, not a medicine. During World War II, on the 2nd of December, 1943, a German air raid sank 17 Allied ships, including the SS John Harvey, with its secret cargo of mustard bombs, in Bari Harbor, Italy. More than 1,000 men plunged into the blazing oil-covered sea. Those who survived suffered the severe, toxic effects of nitrogen mustard. 2 American heroes emerged from this tragedy. Dr. Stuart Alexander, who first observed and reported the profound and specific effects of nitrogen mustard on the immune cells of the sailors. The second was Dr. Cornelius Rhodes, a brilliant pathologist, oncologist, researcher, and chief of the Chemical Warfare Service. Dr. Rhodes recognized the hidden potential of a wartime poison gas, and its immense benefits, as a treatment for leukemia and other cancers. After the war, he became the first director of Sloan Kettering Cancer Institute, and established the multidisciplinary team approach to fight cancer. His pioneering work ushered in the era of modern chemotherapy in 1946. For the next 60 years, new categories of anti-cancer drugs were developed and tested by pioneering physicians and scientists, to include anti-metabolites, anti-tumor antibiotics, plant alkaloids, platinum agents, antitubulin agents, and monoclonal antibodies against tumor cell receptors and blood vessels. In 2011, a new anti-cancer drug, a type of immunotherapy, was approved by the FDA, making world history. Chemotherapy has made tremendous strides over the last 60 years. Chemotherapy targets and kills cancer cells by damaging DNA, or preventing cell division. However, cancer cells outsmart our drugs by mutating, resisting, and suppressing our immunity. Cancer immunotherapy is fundamentally different. Instead, it unleashes our own killer T cells, sending them off on a seek and destroy mission against cancer cells. It is like unleashing a bulldog on an intruder at your door. This incredible breakthrough took 4 decades. Today, we welcome and celebrate the brilliant, tenacious scientist who devoted his life to studying T cells, when very little was known in the 1970s. He fought against all odds to develop an anti-cancer drug to cure cancer. Dr. James Allison was born in Alice, Texas. He studied at the University of Texas in Austin, where he received his doctorate in biology in 1973. He then worked at several academic institutions, to include the Scripps Clinic and Research Foundation in California, University of Texas Cancer Center in Smithville, Texas, UC Berkeley, and Memorial Sloan Kettering Cancer Center in New York. Since 2012, he has been a professor and chair of immunology at MD Anderson Cancer Center. His first major achievement in immunology in the 1980s was identifying the T-cell receptor, which he calls the ignition of the T-cell. In 1992, he showed how CD28, another molecule on the T-cell, functioned like the gas pedal of a car. Then, in 1995, he identified another molecule called CTLA4, which he calls the brakes of the T-cell. By taking the foot off the brakes of T-cells, using monoclonal antibodies, T-cells were unleashed to attack cancer cells. After seeing how tumors on his mice shrank and disappeared post-treatment, he knew his antibodies worked. After a decade of struggle with drug development, big pharma and clinical trials, Dr. Allison's drug, ipilimumab, was finally approved by the FDA in 2011. In 2018, the Nobel Committee awarded Dr. Allison the Nobel Prize in Medicine, in recognition for discovering the immune system's role in defeating cancer. The 2019 film, Breakthrough, is an inspirational, must-see documentary about one warm-hearted, tenacious man's visionary quest, and struggles to find a cure for cancer. Here's the 2-minute trailer.
Part of Jim's success is the fact that he's an iconoclast. Great scientists are creative people.
Jim's lab has a feel of being like a pirate ship.
Jim theorized that tumors have evolved to trick the immune system.
At the time, it was a crazy idea.
This constituted a real breakthrough.
I knew that this is a way to cure cancer, and I'll be damned if I'm going to let that just lay around.
For the next decade, he had one goal, to get that made into a drug for cancer patients.
It was just no, place after place after place.
Many of my colleagues said, this will never work.
My mother sat on the bed with her and held her hand while she died. I found out later that it was lymphoma. When I was first told I had prostate cancer, I was terrified because it was a week after my brother had died.
BMS was not sure they were going to take it at all.
I must admit I was apprehensive.
His experiment in this was to just keep fighting, to say, there's something here.
He's willing to do it in the face of his colleagues not necessarily believing him.
He wanted to stay on the scientists, he wanted to stay on the FDA, he wanted to stay on Bristol Myers Squibb.
I need to get it into people. I need to get it into patients.
I got a phone call from the radiologist. He goes, what did she get? All the cancer went away.
Until then, it was just a concept. She was the first patient I'd ever met.
How in the world are you supposed to adequately thank somebody that without them, you wouldn't be here?
That makes it all worthwhile.
It takes a really powerful idea combined with someone willing to push it forward to make it happen.
The number one thing I learned was how important it is just to persevere.
We knew we were changing the world.
I asked you to step forward to receive the Nobel Prize.
Without further ado, please welcome our keynote speaker, Dr. James Allison.
Sir, it's all yours now, so thank you.
Okay. Okay, well, everybody, sorry for the delay. There are my disclosures. Next slide. Next slide, please. Yeah, so what I'd like to do today is talk to you about checkpoint blockade, specifically in cancer therapy. Start with a little bit of history with some of the fundamental immunological observations that we made that this whole thing is based on, sort of bring you up to date where we are now in treating a variety of different types of cancer. And then finally, at the end, just show you some examples of things that we've been doing here at MD Anderson to try to make checkpoint combinations, new combinations to make checkpoint blockade work better. And then speculate a little bit about where we're going in the future. Next slide shows some of the collaborators. But I just want to stress that this work was done, some of it by myself, but a lot of it by Padmini Sharma, who, and since transparency, actually is my partner, but also my wife, in a laboratory that we run at MD Anderson called the Immunotherapy platform, in which she's the scientific director. Next slide just shows you that this really does work. I think in the most of you saw in the clips that were shown before we started early this morning. This is of Sharon Belvin, the patient that was talked about there. She was diagnosed when she was 22, just out of college, just got engaged to get married, just about to start her job. Anyway, she was diagnosed with stage 4 melanoma. She had 31 lung mets. You can see some of them circled there on the left panel. She had already failed 2 different chemotherapies, high-dose IL-2, a dendritic cell vaccine, and also radiation for a brain met. She got a single round of anti-CTLA-4, and as shown on the right, her tumors completely disappeared, her lung nodules completely disappeared. And the next slide shows that also, you can see before treatment, that brain met at the lower right on the left panel, but that also resolved completely. This was in 2000, this was about 2006, I think, when these were actually made after she finally got to the therapy. It's shown on the next slide, just a photo made of her with her 2 children in 2016. And they're still doing fine. And next slide, I added this during the interim here while we were talking because she and I have become pretty close friends. And anyway, she went with me to Stockholm for the Nobel Prize. And, she does a lot of promotional speaking and talking about, cancer therapies and helping patients realize that, there are alternatives available. Next slide then just shows you what this is based on. It was also mentioned in one of those earlier films when I was working at Smithville for MD Anderson in the 70s, early 80s. In 81, we published the protein structure of the T-cell antigen receptor, which is like the ignition switch in the car, and made antibodies to it. And that sort of opened the door for a lot of work, including the cloning of the genes by others. But very quickly, the field realized that it's not that simple. You can't just flip that switch and get T cells activated. There's another signal, what's called a co-stimulatory signal, that a lot of people showed could only be provided by certain really good antigen-presenting cells called dendritic cells, a specific type of innate myeloid cell that can effectively present antigen. And we showed that the receptor for those signals on T cells was a molecule called CD28. But the structure on antigen-presenting cells is for molecules called B7 or CD80 and CD86. And when those engage CD28 on the same cell, that is seeing antigen and by the T cell antigen receptor, those 2 contemporaneous signals turn on 2 programs in the T cell. The first one we knew about immediately, and that's this shown in green, which is the production of a lot of proteins that are required for cell cycle progression, and turning off things like P27 KIP that inhibit cells from proliferating, turns on anti-apoptotic factors like BCL-XL, which keep T cells alive. And the net effect of all that is IL-2 production, and the T cells expand. This is important because in your body, you probably have somewhere around 100 million different clones of T cells, different specificities. So obviously very small numbers of any one. And when the T cells get activated, that particular clone has to expand to give you the army of hundreds of thousands, if not millions of cells that then swarm through the body and look for the virus, or in this case, the tumor antigens, whatever, and take care of them. That's a critical phase, this expansion phase to generate the soldiers. The army from a few soldiers. It used to be thought that they just died from stimulation, activation-induced cell death was what terminated that, which was never scientifically very, interesting or compelling. But actually we found, right after we cloned and showed the function of CD28, there was another molecule that had been cloned and sequenced by another group called CTLA4. Nobody knew what it did, except that it was turned on after T cells were activated. There was a lot of controversy. It was very homologous to CD28. And so the first group that made antibodies to it concluded that it was another CD28, it was another co-stimulatory molecule. But our data, developed by a graduate student in the lab, Max Krummel, we decided that more supported that it was an inhibitory molecule. And actually, one of the things that we did and others did to prove our hypothesis, which was based on a lot of in vitro experiments in the laboratory, was to knock the gene out. And when we knock CTLA4 out in mice, they die when they're about 3 weeks old because their T cells start dividing and can't stop. So that is the function of CTLA4, which is that second program. The gene is silent. When CD28 and the T cell receptor give their signals, that turns on the CTLA4 program, which ultimately leads to accumulation of CTLA4, which binds the same ligands that CD4 does, but about 10,000 times tighter. And so that actually just stops effective co-stimulation and attenuates and finally terminates the response so that it won't kill you. That's built into every response. The CTLA4 is the brakes on the immune system. Okay, so what does that have to do with cancer? Well, as shown in the next slide, we did a lot of experiments before we knew about CTLA4, where we put B7 molecules into tumors, a large number of different kinds of mouse tumors. And what we found was they get immediately rejected if they express B7. So the reason that they are growing is not because, as many assumed, that they lack antigens, that is, they lack things for the immune system to detect, but it's because they can't give that second signal. Even though they've got the antigens, they're invisible to the immune system until the tumor gets big enough that it perhaps can't get enough oxygen, can't get enough nutrients, whatever. The tumor cells start dying. Next slide. And this causes inflammation. The antigen-presenting cells come in, phagocytize bits of the dying tumor cells, redisplay the tumor antigens on their surface, and by this process called cross-priming, they give the two signals then that for the first time activates the immune response by giving the TCR and CD28 signals. But that also turns on CTLA4. The next slide shows with time, again, CTLA4 will turn that off regardless of what the tumor's doing. So one of the things that I reasoned when we figured these circuits out, I thought, well, maybe since the tumor has a head start, that you could look at it as a race. And if the CTLA4 pathway turns the immune system off to keep from hurting you before it's eliminated all the tumors, then maybe the tumor wins. So let's try to take the brakes off, shown on the next slide, just by giving antibodies to CTLA4 and taking them out of the picture. And so, as I'll show you in a moment, that really works. And there were two reasons why this was compelling when I first thought of it. One of them is that you're treating the immune system, not the cancer. And so the immune system doesn't know if it's a breast cancer or kidney cancer or anything else. It just knows it has things in it, mutations, we know now, because that's what causes cancer, mutations in a variety of cellular regulatory components, and that's where you get cancer. But in the process of developing, you also develop a lot of mutations in other things that all can be detected by the immune system. So there can be multiple targets that you can reveal, again, regardless of the kind of cancer, possibly by blocking CTLA4. And the second thing that was compelling, shown on the next slide, just adding some fill. This happened spontaneously by necrotic death of the tumors, but you can then use a vaccine, or you could get this process going by killing tumor cells with chemotherapy, radiation, hormone, whatever. The standard ways of treating cancer, the conventional agents, can kill tumor cells, but if you don't kill every last tumor cell with those agents, the tumor wins. But because they start by killing cells, that can start priming an immune response. And then by blocking CTLA-4, we could give chemotherapy, we could give radiation, we could give these other therapies some of the properties of the immune system, such as memory. Once you've got T cells, a certain number of them become stem-like cells and are there for the rest of your life, hopefully. And the second thing is that you can reveal T cells, which would detect not only the mutations that are responsible for driving the tumor, but any other mutations in the cell potentially are candidates for drugs. So you can turn one drug, for example, into dozens of drugs, perhaps, depending on the number of mutations in the tumor cell. So for those two reasons, I was really, taken with this idea based on the basic biology of a completely new strategy for treating cancer. Of course, the first question is, does it work? And the next slide shows one of the experiments that we did in mice. This is an experimental tumor growing on the backs of mice, genetically identical mice, and of the same strain that the tumor was derived from. And we did this with a lot of different tumors and a lot of different strains of mice. And basically, very often, just with monotherapy with anti-CTLA-4, we could get the tumor ejected. But what you see here is an untreated mouse in the black curve, are treated with a control antibody. But in the green curve, what you see are the size of the tumors increasing with time of mice that were injected with antibodies that block co-stimulation through CD28. And you can see the tumors grow a little bit faster, showing that there is a bit of an immune response, even if you don't do anything about CTLA-4, but it's not sufficient of taking it out. But if we inject anti-CTLA-4 antibodies, the tumors would grow for a little while, and then the tumor would be rejected. And for the rest of that life of that mouse, they would reject that tumor if we gave it to them again. Now this worked, we found that it did work in all tumors. It worked in some, those that were considered to be highly immunogenic and, for example, were caused by carcinogens. But some spontaneous tumors didn't respond so well. But if we added chemotherapy or a little radiation, or in some cases a vaccine, we didn't find a single mouse tumor that we couldn't cure by this sort of approach. And so after a lot of coaxing and a lot of rejection, finally I convinced a little company called Medarex, shown on the next slide. Without going into all the details, Medarex was a company that had made mice that had their antibody genes removed and replaced by humans. So we very quickly, with Medarex, made a fully human antibody to CTLA-4, which could go directly into patients without the need to be humanized, as most antibodies do. And so the early studies done by Medarex showed that there were responses, objective responses, in many types of tumor, including melanoma, prostate, kidney, bladder, ovarian, lung, just as we would have predicted. Not everywhere. Pancreatic cancer and glioblastomas did not respond and still don't very well, which is something we're working on. But still, many different tumor types. There were adverse events that we had not anticipated because we never saw those in mice or even in monkeys. But they're typically colitis, hepatitis, apophysitis, pneumonitis, inflammatory diseases. They're serious. They can be fatal. But now, just within any new treatment, there are algorithms available that are for treating them. They're generally manageable with systemic steroids, and the patients can be weaned off the steroids, and they don't come back. So these are really just temporary inflammatory disorders. They are immune-based, but they're not really autoimmunity. But we now know with in some patients there are cases where it looks as if there was induction of type 1 diabetes, for example, although the amount of patients might have had subclinical levels. And also a T-cell mediated myocarditis, which can often be fatal because it results in heart attacks. But they're being algorithms developed to detect that early, and we've developed a mouse model of that with which we could test treatment regimens. So problems, very serious problems there, but they're quite rare, 1% or less. To show the next slide, after a while, Bristol Myers Squibb joined the team and took this up to a phase 3 trial, and it was pretty hectic going. They started the trial with the endpoint of progression-free survival, of the RECIST criteria, where progression-free survival was the endpoint, which we told them was dangerous because in the mice, the tumors generally progressed before they went away. That's the biology of this. This is not a drug that kills tumor cells, and you can expect them to just disappear instantly. You've got to build that army. And so after a while of seeing examples of patients that progressed and then their tumors went away maybe 4 months later or 6 months later, they changed the endpoint to overall survival. So the trial took almost 5 years instead of 18 months as it was anticipated to do. It cost, as you can imagine, a lot more. In any event, this shows the Kaplan-Meier curve, the survival plot, where you can see the lower line is a placebo control, which in those days was not unethical because there was no standard of care for melanoma. The median survival after diagnosis at the time was actually 7 months, with fewer than 3% of patients being alive at 5 years. The top line here, the dashed one, is actually the ipilimumab only curve. And what you can see is there's about 4 months or so increase in median survival, which would have been sufficient for FDA approval because no drug of any type had ever prolonged survival with melanoma in a randomized trial. But the really cool thing about it is that you can see that the survival curve flattens out at about 20%, a little over 2 years, and stays there for the duration of this trial. In any event, it was approved by the FDA in 2011, the first immuno-oncology, to use the new name for it, of the modern era to be approved by the FDA. It's shown in the next slide, which sometimes take there. Good. This is a 10-year follow-up of almost 5,000 patients, and what you can see is that inflection point at about 2 to 3 years is real, and very few patients die after that point. So about 20 to 22% of patients were alive in this study 10 years after therapy finished. And so I think that was quite exciting at the time because, as I said, it was pretty much uniformly lethal. But to be able, and this is why in that film I think that we can start using the word cure for patients who have been out that long. And so pretty satisfying accomplishment. But why 20%? Why isn't it 100%? Why doesn't everybody respond? Well, there are a couple of mechanistic reasons I could suggest, but of course the most interesting one to a scientist, and maybe obvious, is maybe CTLA-4 is not the only checkpoint. Maybe there are additional ones. And so about the time our initial clinical data started coming out, Tasuku Honjo and his collaborators, who had been working on a molecule called PD-1 for several years, but they didn't really know what it did, turned out, shown on the next slide, Honjo and Gordon Freeman and Arlene Sharp, Honjo from Japan and Gordon Freeman and Arlene Sharp at the Dana-Farber Cancer Institute, showed indeed that PD-1 was another checkpoint. Very different, though. One of its ligands is actually expressed on tumor cells, PD-L1, and it's different because it's expressed in response to gamma interferon made by immune cells. And so this is an acquired resistance mechanism. The T cell tries to attack the tumor cell, CD8 T cells, for example, by recognizing antigens on their surface. They make gamma interferon, and the tumors then upregulate PD-L1, which turns the T cells off and keeps them from dividing. And so that's how they defend themselves. So it works very differently than CTLA-4. PD-1's real biological role probably is in protecting the developing fetus against attack by the maternal immune system against paternally encoded proteins. And tumors seem to have co-opted that to protect themselves. But anyway, very different mechanism, which operates not during priming, but on fully differentiated cells. And I don't have time to go into it today, but that makes a fundamental difference in how they work and some of the things that they do, which again, I won't have time to go into today. But in any event, very quickly, human antibodies to PD-1 were made, went into clinic. Shown in the next slide are just the results from a phase 1 trial by Suzanne Topalian at Johns Hopkins showing good response rates in melanoma, non-small cell lung cancer, renal cancer, no response in colorectal cancer, although it was quickly found that there is a subset of colorectal cancer with defects in DNA damage repair or microsatellite instability. They have a lot of mutations, and those patients respond quite well. Also, there were no responses in castrate-resistant prostate cancer. And later on in the talk, I'm going to show you why that is, why PD-1 doesn't work against it and CTLA-4 does. That's one of the things we've been working on since then. Anyway, the next slide just shows that very quickly it was shown that these seem to be mechanistically distinct, and they are, because patients that progress on anti-CTLA-4 on IPI do respond to anti-PD-1 nivolumab at essentially normal rates, and the reverse is also true. Those that respond that fail and progress on Nivo PD-1 can respond to anti-CTLA-4. And this, of course, suggested to us that since they're independent, maybe you could put them together. And the next slide just shows some experiments that were done by putting them together. Next slide, by Mike Curran in my lab in a mouse model using anti-PD-1 or PD-1 ligand or anti-CTLA-4, or all three together, showing that there's at least an additive contribution of these because they are distinct mechanisms. This then led to Jed Walchuk, who you met earlier in the film, and who was Sharon's good colleague and personal friend of mine, who's also Sharon Belvin's doctor. Anyway, he designed a series of combination trials in melanoma. Shown in the next slide, I'll just show you one example of this, where again the IPI, the CTLA-4 response rates, the survival rate is a little above 20%. But when you add PD-1 to it, it's in this slide, it's about 55%. Now this just shows about 18 months of follow-up, but now there's 5 years of follow-up. I don't have a curve showing that, but it's the patients from the phase 3 randomized trial, and about 55% of those patients are still alive after 5 years. And so there's no reason at all to expect that they won't be alive in 10 years or 15 years. Actually, the oldest, the person that's the farthest out on IPI right now is 19 years out from the first phase 1 trial. In any event, there's no reason, I don't think, although the data is just for 5, but why this combination won't be curing over half of melanoma patients. So gone quite a long way in melanoma since the days when it was considered a fatal disease and there were no FDA-approved drugs to treat it. The next slide just shows one of the things that became pretty clear pretty fast, and that is that tumors really fall into 2 categories. Those such as melanoma, shown here to the far right, respond quite well. These, of course, are a result of mutations caused by ultraviolet damage to DNA, and the very large tumor mutational burdens in melanoma. But also there are other cancers such as associated with carcinogens found in smoking, such as lung, stomach, esophageal, bladder, etc. all of those kinds of cancer respond very well to monotherapy, actually either with PD-1 or CTLA-4. But when you get down to other kinds of cancer whose etiology is not so clearly associated with mutagens, both the mutation rate and the response rate to anti-CTLA-4 falls off pretty quickly, at least to monotherapy. Kidney is an example in the red squares there. It responds quite well for reasons that we don't quite understand. But if you go further left, you can see that pancreatic cancer and glioblastoma don't respond. And as I told you, prostate responded to anti-CTLA-4, but not to PD-1. And again, we'll come back to that in a minute. But just to go forward, next slide. We know now, next slide, and this is by no means a complete list. I didn't have much room for all the approvals. Now there are 48, I think, approvals for different indications, specific indications. There are 7 antibodies to PD-1 in development, 2 different ones to anti-CTLA-4. But again, IPI was the first in melanoma, and then PD-1, and then the combination. But now, and I'm not going to go through it in detail, but in lung, renal, colorectal, head and neck, et cetera, a lot of tumors respond. What's happening now is, and I should point out, there's also one very interesting approval for one of the PD-1 drugs, which is approved for any kind of cancer, regardless of histotype, that has defects in DNA damage and consequently has high mutational burdens. There is a fraction of tumors in almost every kind of cancer like that are called hypermutators, and regardless of histotype, it's approved to use PD-1 drugs at least to treat that. So very, very interesting thing. But what's happening now is that the combination of anti-CTLA-4 plus PD-1 is moving ahead as sort of the core treatment in most of these because of the efficacy of that combination. The limitations, as I'll tell you in a minute, are that that's somewhat more toxic than the monotherapies, but I think can be managed. The next slide just shows you sort of an introduction to what I'm going to switch over to now, and that is the immunotherapy platform at MD Anderson. So this is based on some work started by Dr. Sharma. She's a GU oncologist and has a PhD in immunology. But back in 2006, before anything was approved, when we had very little insight into the mechanisms in patients, she did a pre-surgical trial in bladder cancer. It was sort of the model for learning how to look at mechanisms, and that gave plenty of tumor tissue and blood to study and look for markers. I don't have time to go in detail through what she found, but one of the things she found was that CD4 cells expressing ICOS were a pharmacodynamic marker that predict when CTLA-4 antibodies, for example, have hit their target. They don't really tell you who's going to respond. They'll tell you who's not going to respond, because if you don't see appearance of those cells, they're not going to. Any event, what that sort of ushered in is this idea that she had that we've worked on together, which is do clinical trials, small clinical trials, maybe 12 to 20 patients, but biopsy intents, get tissues, get blood, take them back to the laboratory, study them, see what happened, generate sort of in a reverse translation, generate hypotheses of how it's working, which you can then test in mouse models using knockout mice or combinations of antibodies or whatever. And since we started this, when I arrived at MD Anderson, we've got over 4,000 patients now that we've enrolled into a blanket protocol that lets us take samples from any patient receiving any therapy at MD Anderson that might impact the immune system. And so we get longitudinal blood, longitudinal biopsy specimens, and again, study them in the laboratory. So again, I'm going to give you several examples of what we've been doing there. The next slide just shows you one of the things that is a problem, and that's cold tumors. There's two meanings of cold versus hot in cancer. One of them is mutational burden, but the other one is immunological. And here we talk about cold versus hot, meaning, how infiltrated are they with immune cells? And here, unfortunately, they're not labeled all that well, but it's pancreatic is on the left of each of these boxes, prostate the next one, bladder the next one, and then melanoma to the right. So what you can see is that melanoma has very high levels of CD8 T cells in them untreated as compared to the other types of cancer. There are immune cells, however, detected by CD45R0 in melanoma and in bladder and in pancreas and in a little bit in prostate cancer, but it's really, and then also granzyme B, which are activated CD8s. But really, pancreatic and prostate are not that different. They're both cold with respect to the genetics and cold with respect to infiltration, which is poses special problems. Shown on the next slide, because you've got to get T cells in. A hot tumor like melanoma or even bladder or the kinds that I talked about earlier that are associated with smoking have a lot of mutations, but also tend to be pretty heavily infiltrated. So all you have to do is release the checkpoints, and you could unleash the immune system to attack them. But in cold tumors, you've got two challenges. One is to get the T cells in and the other immune cells in that you need to mount a successive thing. And then, and only then, can you release the checkpoint. And so, the next slide, let's just go back to prostate cancer. As I told you, there were responders, even in the phase 1, the early probing studies of trials, small trials that were done by Medarex. Using ipilimumab. That resulted in a couple of phase 3 trials of ipilimumab in castrate-resistant prostate cancer, one of which is shown here. The other one's essentially the same, but not really a convincing effect overall. Some responders, but it failed to reach any level of significance much better than the standard of care. So those were failures. And that, together with the fact that in the early trials that BMS did with PD-1 antibodies, where there were no responses whatsoever with PD-1, they shut down their prostate cancer program. But shown the next slide, based on the principles that Pam Sharma had developed, we decided to go ahead and do a pre-surgical trial with IPI and hormone therapy in prostate cancer. And so what I'm going to show you, this is a localized disease, actually. And I'm not going to show you all the data because it turned out that the hormone therapy didn't really contribute much. It didn't contribute at all from what we could tell to the immunological changes going on. But shown on the next slide, the patients that got a couple of doses of IPI before they went to surgery, as you can see on the left of these panels, these are slides, the pre-treatment samples were very cold, very few CD4 cells, CD8 cells, FOXP3 regulatory cells, or immune cells in general. These ICOS positive CD4 cells that we found that correlated with ipilimumab treatment were not there. Neither were many granzyme B expressing CD8s or CD68, which marks myeloid cells, macrophages. There were some macrophages, but not many. But after treatment, look at the right side with anti-CTLA-4, you can see robust T cell infiltration, some regulatory, both CD4 and CD8. You see appearance of these ICOS positive CD4 cells, a lot more granzyme positive cell, and a lot more macrophages. And that's why CTLA-4 worked to a point, because it has the ability to drive T cells into a tumor, whereas PD-1, and we've got similar data for renal cancer as well. So at least to those two cold tumor types, the CTLA-4 can drive T cells in, whereas PD-1 monotherapy does not. But one of the things we noticed right away, shown on the next slide, when we did an expression analysis, looking at RNA for induction of new genes, is shown on the right. The next slide, just highlighted on the next slide. PD-L1 VISTA, sorry, go back one. Circled in red are PD-L1, the ligand for PD-1, also VISTA, PD-1 itself, and then LAG3, which are additional checkpoints, additional molecules that can inhibit T cell. So when you treat with anti-CTLA-4, one of the things that we know now is a general sort of phenomena. The immune system responds by putting up other checkpoints to try to, bring the system back into control. And that's why we decided that PD-1 might not have worked. CTLA-4 gets the T cells in, but now you've got these new suppressive molecules that weren't there before. Shown on the next slide now. Go ahead. Again, pre-treatment, there's no PD-1, no PD-L1, no VISTA, this new marker. But after treatment, a lot of PD-1 on the immune cells, PD-L1 on some of the immune cells, and this VISTA molecule, it's a macrophage. And then the right shows on tumor cells, robust PD-L1 expression now by the tumor cells. And these data are quantitated on the next slide, just to show you that after treatment, there's CTLA-4 on the tumor cells, on a subpopulation of macrophages, and on a subpopulation of even the T cells themselves. So the reason that CTLA-4 didn't work was because it drove T cells in, but it induced these ligands. The reason that PD-1 didn't work was because there were no T cells in there in the first place. And so we thought that, well, why not think about putting them together? So we show on the next slide, again, you look at VISTA, and also VISTA after anti-CTLA-4 treatment is expressed a little bit by T cells, but on a big subpopulation of myeloid cells, which incidentally is, interestingly, never on the same cells as PD-L1 on myeloid cells. So there's two different myeloid cells that get brought into inhibitory functions by treatment with anti-CTLA-4. So then we, next slide, went to BMS and showed them these data and said, what we need to do is give IPI to get the T cells in and PD-1 to take those molecules, those inhibitory molecules of the PD-1 axis out of the way. And maybe we could add VISTA later on if we need to get it out of the way too. So they reactivated their prostate program and gave us some antibody to do the combination therapy in patients with melanoma. With prostate cancer, with castrate-resistant prostate cancer. So in the next slide, it just shows one patient. His PSA level was going up. You can see the red arrow. The first dose of the combination therapy was at the nadir of the PSA, which started down, then finally reached undetectable after the third dose. The patient was a complete responder. All the soft tissue metastases went away. And there were several responders like that in this trial. Bone mets didn't respond, and I'll come back to that in a moment. But the soft tissue mets and the prostate itself, the tumors completely resolved, and the PSA, as I said, became undetectable. There were problems with toxicity, however, because these prostate cancers are generally patients are generally older and sicker than melanoma patients. Shown as the next slide is just a schematic for the trial. We carried out the data that I showed you was from the signal-seeking study. It was 90 patients, and then this was expanded now to a multi-institutional, very large, hopefully registrational trial with smaller doses or bigger spacing between the doses of ipilimumab to try to get around the toxicity. And so that's underway now. And again, it's based on our data from the immunotherapy platform. And, we're optimistic, but the data we'll have in a couple of years. And hopefully that'll work. Show the next slide to go just sort of continue. So what I showed you is you could make the cold tumor hot. You can get T cells into prostate cancer, but are there antigens there? Because the dogma, in the field now has become that, higher mutational tumors with a higher mutational load are a lot more likely to respond to checkpoint blockade than those with a lower mutational burden. And that's true. It makes a lot of sense, because not all mutations are going to be recognized by the immune system. It depends on your HLA type, whether they actually get presented. But many of them will, and the more mutations you have, the more shots on goal you have, basically. But prostate is notoriously cold, again, colder even than pancreatic in terms of mutation burden. So what we decided to do, show the next slide, was to take a panel of patients that have metastatic prostate cancer, resect their primary tumor, do exome sequencing and compare it to blood to define the mutations that are there, do RNA sequencing to find those that are actually expressed at the RNA level, and then give us an idea of the peptides that might be derived from the mutations in the tumor that we could study in vitro, the laboratory responses. Seem to have lost. Okay, there we are back. But ongoing responses even after 2 years. So these are very solid responders. The next slide, just to concentrate on those two patients. Well, it shows the mutational burden, but as you can see here, there are a few patients that have number 4 has 700 mutations, number 15, 104 mutations. Those both had an atypical small cell prostate cancer that's very difficult to treat with anything. Most of the patients fell into a much lower range with in the case of expressed mutations in the far right column, that patient 5, who was a good responder, had only 12 mutations that were actually expressed at the RNA level, and number 7 had only 10. And so, the expectation would be that maybe neither of those would be detected by T cells in the patient. So, shown the next slide, though, we could detect with time using what's called an ELISpot assay, where we take T cells, we look at the wild type peptide, the unmutated one, and then irrelevant peptides as negative controls, and then a cocktail, which are on the bottom in all these, and then a positive control, which at the top, which you can see of each of these figures, which is a cocktail of peptides derived from common viruses that everybody's had. What you can see here is one mutation in patient 7 who had, actually this guy does have 2, sorry. One of them in a rho guanine nucleotide exchange factor 37, who has a point mutation, is shown in that box at the red. The wild type peptide evoked no immune response, but the mutant peptide, there are an increasing number of T cells with time, as you can see here by the increasing number of dots on the plates labeled mute. They're quantified there. So there weren't any T cells until after the ipilimumab therapy, but both that mutated protein, and the second one called dihydropropyl minidase on the right, again, another mutation where the T cells didn't detect the wild type protein, but did detect the mutated protein. And same was true of patient 5, who, go to the next slide. Actually, I took that out, but it was the same thing, except there was only one. But anyway, both these patients who were responders had robust T cell responses to these mutations induced by the therapy. As shown on this slide, however, they also had T cells that protect to known shared antigens in prostate cancer. On the left, prostatic acid phosphatase. In the middle, prostate-specific membrane antigen, and to the right, PSA itself. But the PSA, there were T cells against PSA even in non-responders, so that we don't consider to be very significant of anything. But this, of course, complicates our ability to attribute the cures, or I should say cures because these patients have not been there, to these responses, though, to the mutated peptide, because there are these shared antigens. But at least it showed that you don't need hundreds of mutations to get responses, T cell responses, that just a few may be sufficient. So there is hope for other kinds of cancer with low mutational burdens. Okay, the next slide deals with another issue, and that is, I told you the soft bone metastases responded to check, the soft tissue metastases responded, but the bone metastases did not. And of course, that's what kills most patients with metastatic prostate cancer. So we decided to have a look at that and see why is that. And so this just shows on the left here, these are gene expression arrays, and the top is Th1 associated. These are T cells that make gamma interferon TNF alpha and can kill tumors. Th17 genes in the middle panel. These are associated usually with adverse events and allergic reactions. And at the bottom are Tregs. These are inhibitory T cells. What you can see is soft tissues. Pre-IPI, you don't see much of these. But after treatment with anti-CTLA-4, you do get in soft tissues or in the prostate these Th1 responses, some Th17 and some Tregs. But on the right are bone mets from several patients. And so basically they as I said, they didn't respond. But if you look at what's going on there, just at the genes that are expressed, again, even after therapy in bone mets, there's no Th1. What you tend to get is a Th17 response, and again, the Tregs don't really seem to matter. But as you can see from all of those sample patients there, even after treatment with anti-CTLA-4, I mean, there is a response, but it's Th17, which has no good. That does not do anything to tumors. You need a Th1 response. So why is that? And so, again, consistent with our philosophy of reverse translation, as the next slide, we developed a mouse model developed on the basis of these data, where we just take mice and gave them a prostate cancer cell line injected either into soft tissues or into the bone. And what you can see here is looked at CD4 T cell subsets in the bone, as shown on the left. We tend to get the Th17 profile, not a Th1, but in the soft tissues, you can get, after treatment with anti-CTLA-4 again, which is the red symbols, you get more of a Th1 in the soft tissues than a Th17. Now, why is that? So next slide. So this mimics the human situation. There are two cytokines which tend to shift cells towards the Th17 phenotype. It's interleukin-6 and TGF-beta. And as you can see here, our studies in the mouse show that those cytokines are made in the bone marrow, whether there's a tumor or not, and especially if you have a tumor in there. So, and then those, we think, are the things that shift it towards a Th17 response. So the next slide just shows that data developed showing that if your IL-6 didn't work so well in the red, so this is anti-CTLA-4 plus anti-PD-1, and just that combination in the gold, if you will, spots, and with IL-6 in the red. But if you add anti-TGF-beta, shown in the green, there were very good responses in the bone mets as well. And so this then led us to say that the location of the metastasis is going to have a lot to say about the nature of the immune response that you're going to be able to induce there. And in the bone, it's very different than it is in other tissues, because these two cytokines, IL-6 and TGF-beta, just divert the T cells down a pathway, which is not very useful to anti-tumor immunity. And you can interrupt that by getting in the way at least of TGF-beta. And shown on the next slide, we've planned a trial then, and this also shows in those mouse models that we expanded gamma interferon producing Th1 cells, Th1 CD4 cells, as well as gamma interferon producing and killing CD8 T cells in the mouse model. If you use a combination of immune checkpoint blockade and anti-TGF-beta. So the principle is shown there by our mouse model. And the next slide just shows on the basis of that, we've got a similar trial planned to use that combination in patients with castrate-resistant prostate cancer. And we'll see how that works when we get it started. And so the next slide, then I think that's all the data. But this is a partial list of the checkpoints that have been identified now. There's several of them. Still, my experience and what I've seen in both animal models and the clinic tells me that you can block multiple ones. At least CTLA-4 and PD-1 are the dominant ones, but but the immune system has a lot of functions, does a lot of things. And so there are many checkpoints that work at many levels of the immune system. And I think it's going to be likely, like VISTA, for example, we might want to add that to the PD-1 and CTLA-4 combination in prostate cancer, because we know in prostate cancer that VISTA molecule is expressed. That wouldn't be appropriate in melanoma, for example, because we can't find VISTA in melanoma, just in prostate. And so there are some positive molecules too. One of them was ICOS, I talked briefly about, that's induced by CTLA-4 on CD4 cells. We have shown in mouse models that an agonistic signal through ICOS can make those CD4 cells work much better in response to CTLA-4. So you can combine, there's in my declarations approach, we made an antibody to ICOS, which is in clinical development now by Jalance company in trials, we'll see. But in mouse, at least, you could combine positive checkpoints, you combine negative checkpoints. And I think the future is that we're going to start combining them in a way that's going to be partially, at least, determined on studies like I've shown you, where you look after treatment at biopsies in patients with different kinds of cancer and look at metastases in different locations. You get the information on what's going on, and base their treatment strategy based on data and not just assuming that that there's any one combination is going to work for all cancers. Although, again, it's moving towards PD-1 and CTLA-4 being the main combination. But even then, in some cancers, the response rate's less than 50%, and we've got to do better. So the next slide just shows again what I think is going to happen. I mean, again, we're going to start combining multiple checkpoints, negative and positive. And as I alluded to early on, I think one of the most powerful things that we could do now is to start combining immuno-oncology techniques with conventional therapies. Because if we can give the checkpoint blockers when we know there's tumor cell death, and we know that priming's happening, and we know that that's the best time to give anti-CTLA-4. If you give it without that, you're just guessing and hoping that somewhere in the body there's some priming going on. But if you've got a good therapy that can kill a bunch of tumor cells, the problem with the conventional therapies is unless you kill every last tumor cell, the tumor's going to come back. And there are, as shown in that really nice the film about the history of cancer therapies, the genomic instability of tumors makes it very likely that the tumor is going to continue to be able to work its way around any conventional therapies, so you'll never kill that last tumor cell. There'll be variants in there that you can't kill. And that's true of genomically targeted therapies as well. So when we start realizing that you can use conventional therapies, as long as you say, let's not try to kill that last tumor cell. Let's use them in smaller amounts and let the immune system do the heavy lifting here and give us the diversity of the response and give us the memory that we need to cure cancer. And then I think we're going to really start seeing the bang for our buck. In any event, the next slide, just to close, I think that what we've done with, since the advent of chemotherapy and radiation, especially, oncology has really proceeded by doing fairly large randomized studies where you take a statistical look at the median survival, and if it's more than a few weeks or months, that there's something to be said for giving patients, as long as quality of life is good, that increase, even if it's not curative. But we know from ipilimumab, shown on the next slide, that you can do that, but you can also get this tail on the curve of 20% or so decade-long, 15, 19-year-long responses that I think can be considered cures. And my final slide, and the one I want to leave you with, and this is not data, this is an aspirational slide to say, rather than concentrate on the median survival, we need to be concentrating more on that tail on the survival curve and raising that as high as we can get it, as many different kinds as we can get it. As I say, we're not there yet. We're at 55% perhaps for metastatic melanoma, lower for other kinds of cancer. And through the sorts of approaches that I've described to you. I think that we'll be able to do it for many kinds of cancer. Not all, perhaps. I don't think the human race will ever be totally free of cancer. We haven't had much luck so far, although there is some glimmers of hope in pancreatic cancer, and even a little bit in glioblastoma, I think, but a lot more work to be done there. And so that's what I leave you with, I think, is hopefully an optimistic view of where we're going with cancer therapy, to being able to treat the majority of cancer for the majority of as many patients as we can help. And with that, I'll stop, and I appreciate everybody's patience through the difficulties in getting started. And I don't know if there's a question period or not, but anyway, it was a pleasure to have this opportunity to talk with all of you.
Dr. Allison, we want to thank you so much for that wonderful presentation. Your life's work in immunology has brought hope to so many people around the world, and we just can't thank you enough for 40 years of research. I think we compiled a small list of questions. So I'm going to let Colonel Ager ask the first one. Actually, he will be reading from a list.
Okay.
Colonel Ager, you're on.
Sir, thank you for that great talk. As Colonel Shen-Gunther said, we compiled a list of questions from various folks in our lab, and so I'll just start with one. You're an inspiration to young and old doctors and scientists around the world. What would you say to influence young physicians to encourage them to engage in clinical trials?
I would say that, if you're driven by a passion to understand anything, to understand how things work, including treating disease, I think that we could do all the mouse models we want. I'm a firm believer in mouse models, but you've got to get into humans and see what happens, as soon as you can. I think that it needs to be done a little bit differently than it's done now. I think going for a 600 patient trial until you understand mechanisms in patients is probably a mistake, because you're wasting patients and you're wasting effort. I think that it would be useful to do small trials, lots of small trials, as I've described here, where you look at 10, 12, probably no more than 20. You don't need a p-value. You need to just have insight into mechanisms that are involved that can help you decide if a combination has a chance of working. I mean, we know what a successful therapy looks like, and we know the things that can get in the way. So I would say to be creative. If you've got an idea for a combination, by all means, the way to approach it is to try animal models, but then as quickly as you can, go to, once you've got safety taken care of, go to small trials and just see what's happening, and inform yourself of that, and that'll help you take it forward or improve it.
Thank you, Dr. Allison. I'm going to ask one more question. I'm sure that other folks in the audience may want to ask a question as well. But you talked in your video about moving from one institution to another. Just wanted you to comment on how that change in laboratories, so going from Texas to California up to New York, how that affected your research, and can you sort of talk about the academic cultural differences in the environments? Over.
Yeah, that's an interesting question. Yeah, my first job was at Scripps Clinic, and I was as my postdoc, I was trained in biochemistry. I wanted to learn immunology while I was in school at UT Austin. It was when T cells were really discovered. I mean, and shown to be a separate family than B cells. And I was fascinated by that. The professor, Bill Mandy, that taught the immunology course, and he said, there's T cells and B cells. He was an antibody guy. So, but I went in after class and said, well, how do they work? He said, well, probably they're clonal, just like antibody molecules, but I think they're just cells that picked up antibody. But I decided I wanted to learn immunology, but my training was in biochemistry. So I went to an immunology lab, but I got stuck with a project where I just purified MHC molecules because nobody knew what they did at the time. And And my job was to purify them, get protein sequence, and et cetera. So I did that, but my mentor really discouraged me from, model building, as he called it, which I considered understanding, so that wasn't a good experience for me. But when I started my lab at Smithville, I pretty much had free reign. I had to make monoclonal antibodies to tumors, and I mean that was my main task there. It was what I was hired to do in a carcinogenesis setting, to find molecules involved in carcinogenesis and liver cancer. But on the side, I decided I wanted to work on T cell receptors. So that was the holy grail, and so I solved that. And then that got me, and I was isolated at Smithville, and because when I finally surfaced with that and started talking about it publicly, everybody said, "Oh, you can't have done that." There's a group at Harvard working on that, and a group at MIT, and a group at WashU, and they've all got the answer, and which it wasn't what I had. Luckily, one of the things about Smithville was I was isolated, and I didn't have people. I mean, I was ignorant in a good way, and isolated. So I probably wouldn't have undertaken that task, because I would have been told by everybody there were some people way ahead of me. But I got there first, But that got me a job at Berkeley, which of course is a fabulous, fabulous university. Some of the best students and sharpest minds, but it's basic science. It's science at its purest. And there it's so pure that, there was some skepticism about even working on drugs and things. There was, you should be working on first principles, but they tolerated, because we were working on first principles of immunology. But that was where I did, all the stuff, including, the anti-tumor work that we did. And then, it teamed up with Medarex while I was still there. But then when it went into the clinic, I was aware that there was going to be this thing about overall survival versus progression-free survival as an endpoint. I knew nothing about the clinic at the time, but I thought, if I'm going to shepherd my drug all the way through, I better go someplace where I can, do that. So I went to Sloan Kettering, which, is we like to think the #2 cancer center in the world. Actually, anyway, so interesting story, but anyway, so I went there, and as you worked with Jed Wolchok, and was intimately involved. I'm doing the clinical work, obviously, because I'm I'm a PhD, not an MD. But I had the ear of the MDs, at least, and the ear of the advisory boards. And I just kept telling them, don't do RECIST criteria, don't do progression-free survival. It's going to fail. And of course, when Pfizer's drug failed because they did it that way, then BMS finally, said, "Oh, you're right," and switched to overall survival. But I learned about the clinic there, and then I decided to follow that. One of the things that I did find there was that, there's at Sloan Kettering, there's Memorial Hospital and the Sloan Kettering Institute, so there's a bit of a division between the sciences and the clinic, a bit of a division between the the MDs and the PhDs, and that's not true at MD Anderson. At MD Anderson, it's organized in a way that's much more collegial, much more collaborative, and everything, and that's I became aware of Dr. Sharma's work, or Pam's work. I met her at when she was at Sloan Kettering as a postdoc, but she showed me that the early bladder work she did. I thought, this is the way we got to go, and then Anderson offered me the opportunity to start that immunotherapy platform with Dr. Sharma, and so that enabled me finally to realize the part of my dream, which is to make it even better, make immuno-oncology even better through reverse translation, not concentrating on just bench to bedside, but bedside back to the bench, etc. Each one of them. One thing I learned was that anytime you move, there's a sort of repotting thing, rejuvenation, a new outlook, new optimism. But also each of these places that I've been in had a distinct flavor, if you will, that I took advantage of as much as I could. I hope that wasn't too windy.
Well, thank you very much, sir. I see a hand raise. Ms. Wang, would you like to go ahead and ask your question?
Thank you. Thank you very much for a fantastic talk, Dr. Allison. And I have a question that's related to precision medicine. One thing I noticed from your talk is you mentioned the side effects. Some people are less severe than others. I don't know whether it has anything to do with you using the same regime of medicine for all the different people respond differently. I don't know whether you can share with us your thoughts on tailoring the medicine therapy for people.
Well, first of all, I don't think you can't unleash the immune system with, for free. There's a price to be paid in doing that. So, and again, some people, there are people who don't have any side of. I met about a year ago. I met a marathon runner who developed melanoma. I guess for running marathons, I don't know, but had IPI and didn't miss a marathon. I mean, she said she didn't miss a race. But then there's some that have gotten very, very sick. In the early days, there were patients that got colitis, got perforations and died. Not very many, just a few until that was figured out. But now the algorithms are there, but it requires, it's a little bit, the adverse events are more significant with IPI than they are with the PD-1 drugs. That's probably because of the way it works. It works at priming. And so, that, by the way, has a good side, a bad side. IPI, we know now through a lot of mouse work and clinical studies, IPI can recruit new T cells to an immune response against a tumor that weren't there before, whereas PD-1 doesn't do that. It just reawakens the old ones, as you might imagine from the mechanisms that I showed you. But also that means that it's got more side effects. You have to be more careful. So the slogan amongst a lot of clinicians who work with these is you give IPI for durability and you give PD-1 to cut down on toxicity. Well, it doesn't cut it down, but anyway. But so finding the balance between the two, I think, is going to be the important thing about it. But you do have to watch the patients. And one of the things that all the clinicians tell me is that somebody, if not the physician, then somebody that has to have a phone number, a cell phone number. One of the things that I don't have time to get into it, but we've started a little company with a digital way of monitoring over cell phone adverse events. But the thing is that was started because Dr. Sharma had a patient, a renal cancer patient, that she had basically cured, at least, was free of cancer after 5 years of metastatic disease or something. But the guy developed colitis in response to the treatment, but went home and they treated the diarrhea with over-the-counter agents, which of course didn't do anything, and the guy died from the colitis, and not after he was cured of the cancer. And so you have to keep an eye on the patients, and there are ways of intervening, again, with systemic steroids pretty early on, as soon as you start to see something happening, so that you could, hopefully wean them off and keep them on, allow them to finish the therapy. But it just requires monitoring, and they're not stereotypical like they are for a lot of chemo agents or radiation. You don't just give them EPO or whatever. It's got to be tailored to the individual patient. Some of them will, develop skin rashes, others colitis, some pneumonitis, some hepatitis. But most of those are pretty temporary, but you have to pay attention. So.
Thank you, Dr.
Thank you.
Allison.
I see other people have questions. I'll let them ask.
I believe Dr. Bohan has her hand up. Dr. Bohan, would you like to go ahead?
Hello. I really have enjoyed your talk. Thank you so much. I had a genomics question. Mm-hmm. So you had said earlier in your talk that cancers with higher rates of mutations seem to respond better to the checkpoint therapies. Is that all mutations alike, or is it like certain types of SNPs, transitions, transversions, or like indels?
It's things that change, that give you the wrong amino acid in a protein. It could be, several of those, but not necessarily. Those indels won't necessarily do that. We usually think of point mutations, although you can occasionally have a mutation that gives you a new open reading frame, for example, by putting a start codon someplace. It may not normally be even made into a protein, but if you get an in-phase start codon, you can make a protein that wasn't made, for example, or you can read through and get a mutation if you had a tail on something. But typically, it's a point mutation that we're talking about. It has to be something again that changes an amino acid in a protein.
Okay, great. Thank you for clarifying that.
Thank you. Dr. Wade, did you want to go ahead and ask your question that you?
Oh, me? Yes. So, this everything is very trendy now about mRNA therapy for the COVID vaccine, and mRNA obviously can also be used to presumably encode antibodies and whatnot. And do you see in the immunotherapy there is a room for mRNA technology to get into it?
Yes, as a matter of fact, Ugur Sahin, who's the head of he's the guy that made the RNA for the Pfizer vaccine. He started making tumor antigens. And so he had a company. I was in a rival company with a group of people largely from Dana-Farber there. And we both had the same goal, it was to take sequence patients' genomes, find the expressed ones, make vaccines, probably polyvalent vaccines. We considered RNA, but thought, well, peptides are more certain. There's more there. Ugur had the foresight to start thinking about RNA. Anyway, the bottom line is his company bought mine out last year, so he had the better idea. But he moved that to COVID. There's that technology is amazing, and I think absolutely it has a place in tumor antigens. And I'm sure he's working on it through BioIntel or whatever the name of his company is.
Colonel Shen-Gunther, I know that Dr. Allison does have a hard stop coming up, but I think we can ask one more question.
Yeah, sure.
So on the tail of that last question, I wanted to ask if what would you consider some of the most cutting edge or innovative research going on in immunotherapy today, and kind of on the you may have alluded to it already, but who do you follow scientifically? Over.
Oh, okay. Cutting edge, I don't know if I'd exactly call it cutting edge, but I think there's a lot going on identifying checkpoints and things, and that's getting to be a little bit old hat, and I think the returns from that are going to be smaller and smaller. But one of the things that we're realizing in the platform, and I think other people in the country are realizing, that myeloid cells and other cells of the innate immune system need more attention. Myeloid cells, macrophages in particular, their role normally is to help in wound healing. And just so happens, this is a bit of a stretch logically, but that's basically what they do for tumors too. They tend to protect the tumor in a way. And they're myeloid cells which can suppress T cells. And that we're seeing that again and again and again. And many of them do it through checkpoints, but they can also do it other ways, by chemokines. They make cytokines that they make. And we're just beginning to appreciate that. And I think, in our own studies now, I didn't show you, I just showed you a tip of one part of it. We've got a major program now in gene expression, and the other thing about the myeloid cells is they can shift, depending on their environment. They can shift from a pro to a anti-tumor mechanism, and can be shifted. If we can figure out how to do that, I think, and that's one of the big problems actually with glioblastoma and pancreatic cancers, is they have a lot of myeloid cells in them that seem to be suppressive. And right now we're just beginning to grasp how to go about it. So I think that's where a lot of attention is being. As far as a big new quantum leap draft, I don't know. If I did know, I'd be working on it. But I can't. And what was the last question? I forgot. Who do I follow? Oh, I follow a lot of people. Most of them are good buddies of mine in the myeloid area. Miriam Murad at Icahn School of Medicine in New York, or Bob Schreiber at Wash U, Phil Greenberg. Anyway, a bunch of bright people out there. Luckily, I have good luck to be friends with a lot of them. We communicate a lot.
I'm going to turn it over to you, Colonel Shen-Gunther, ma'am.
Sure. Dr. Allison, we want to thank you so much for your presentation, answering our questions. And I know you're a very busy man and you have to leave. So we'll let you go. We sincerely apologize for all the crazy IT issues, but we made it go, made it happen. So
we made it. So
yes.
All else.
Yes, I owe you some fine wine, I think, because of that.
Yeah.
So, all right. Thank you so much, and we'll let you go.
Okay, thank you. And good
Thank
luck
you.
with your work.
Bye-bye.
All of you, good luck with your work.
Thank you, sir. Bye-bye.